Salt Lake City, 1994. A basement office in the Eccles Institute of Human Genetics at the University of Utah. A man named Mark Skolnick is looking at a chromosome map, and the race is over. The gene is real. It is findable. And it was found here, not at Harvard, not at Oxford, not at the Sanger Institute in Cambridge, but here in Utah because of a filing system built by a 19th century church. That's the part nobody leads with. Ray Gesteland, Distinguished Professor Emeritus of Human Genetics at the University of Utah, has said it plainly. More human disease genes have been discovered in the state of Utah than anywhere else on Earth. Not more per capita. More, period. And the reason isn't the equipment. It isn't the funding. It's a database. 8 million people. 23 million linked records. Built from genealogical ledgers that members of the Church of Jesus Christ of Latter-day Saints had been keeping since 1847. Not for science, but for faith. You were told this episode was going to compare Mormon DNA to mainstream American DNA. Here's what that comparison actually found. Genetically, there's almost no difference. The Mormon population's gene frequencies track so closely to their Northern European ancestors that researchers in the 1980s could barely distinguish the two. No genetic isolation. No meaningful drift. No surprise in the genome itself. The surprise is in what that genome built and what building it cost them. This isn't just ancient history. If you have ever received a genetic cancer test, or if someone you love has, then the chain of causation running backward from that result passes through a church record filled out in a prairie settlement sometime around 1850. The test exists because the record existed. The record existed because a community believed that remembering their dead was a sacred obligation. What happened between that belief and that test? That's what this episode is about. If you've ever actually wondered what a community's faith practices can do to the course of human medicine over 2 centuries, subscribe. Because what comes next goes deeper than most people are willing to follow. The name of the community that made more genetic discoveries possible than any institution on Earth does not appear on a single patent. Not one. Actually, before we go further, I want to ask you something. Drop it in the comments before watching this. Did you know that almost every BRCA genetic cancer test in use today traces back to a single religious community's genealogical records? Not to a government lab, not to a university endowment. Right, knew it or didn't know. And while you're there, tell me which side of your family you'd most want to trace genetically if you could go back 5 generations. Because your answer might be more connected to what I'm about to tell you than you'd think. August 1994. The day BRCA1 is confirmed, Mark Skolnick and his colleague Peter Meldrum begin the paperwork to incorporate a company. They call it Myriad Genetics. Its primary asset, its only real asset at the start, is the ability to test for BRCA1 mutations. Within a decade, Myriad will hold monopoly patents on BRCA1 and BRCA2 testing across the United States. By 2013, it will have generated over $4 billion in cumulative revenue. The incorporation documents list founders, they list investors, they list institutional affiliates. The Utah Population Database is not listed. The LDS Church is not listed. The families whose 5-generation pedigrees located the gene are not listed. Now, what made those pedigrees possible? Dr. Geraldine Minot had spent years at the University of Utah building what would become the world's most powerful genealogical research instrument. The database cross-referenced LDS Church records with cancer registry data, death certificates, birth records, 5, 6, 7 generations of family mortality, unbroken, in a population where almost nobody smoked. That last detail, she later explained, was enormous, because when you can remove tobacco from the equation, cancer deaths that are actually genetic start to stand out from the noise. You can see the inheritance. And in the 1970s and 80s, researchers from the university began showing up at something unusual. Mormon family reunions. They were invited. They drew blood. Participation rates hit 60%, 3 times the national average for research subjects. Dr. Loris Betts, then Senior Vice President of the University of Utah Health System, described it exactly. The state population, he said, is a most cooperative group. They had researchers invited to family reunions to draw blood samples. And yet, the data said the exact opposite of what you'd expect a commercialization story to look like. These people weren't passive subjects who didn't know what they were doing. They were enthusiastic, organized, generous. They showed up. They gave blood. They trusted the institution. What they were not told, what nobody told them, was that within 20 years, what they gave at those family reunions would become the foundation of a $4 billion public company. There is a specific mitochondrial haplogroup, K2, found in elevated frequency in the Utah matrilineages selected for the longest lives. Women who carry it die less often of heart disease. They live, on average, significantly longer than women who don't. Their descendants are alive right now. And the women who carried it first, the ones who walked to Utah in 1847, had no idea they were carrying anything. Neither do their great-great-grandchildren. Not yet. Researchers at the University of Utah's Department of Human Genetics pulled up 14,900 distinct matrilineal lines from the Utah Population Database. They ranked them by one criterion. How often did women in these lines survive past 90? Not how healthy they were, not their lifestyle. Just the raw question, who generation after generation kept not dying? What they found was a pattern in the mitochondrial DNA, specifically in haplogroup K, and within that, in a subclade called K2. Mitochondria are the structures inside your cells that produce energy. Most people learn that in school and forget it immediately. Here's the part that matters. K2 carries a specific variant, labeled T9716C, that appears to cause a small inefficiency in the mitochondrial energy production process, a leak. Protons escaping through a gate that should be closed more tightly. This leak reduces the output of reactive oxygen species, the molecules that accumulate over a lifetime and contribute to cellular aging. Less oxidative damage, slower cellular deterioration, longer life. And here's where it gets strange. The same research found that haplogroup K2 was completely absent, 0 for 52, in a group of elite endurance athletes, not underrepresented. Absent. The mitochondrial inefficiency that appears to extend lifespan actively limits peak physical performance. The same mechanism that may be adding years to your grandmother's life is the reason you won't win the Tour de France. This discovery was made in 2018. It barely left the field. Here's what that means for the Mormon longevity story you may have already heard. Active LDS men in California live, on average, 9.8 years longer than comparable U.S. white males. Women live 5.6 years longer. Those figures come from a 25-year UCLA study by James Enstrom and Lester Breslow, published in 2008. The standard explanation is behavioral. No tobacco, no alcohol, strong social bonds, weekly religious attendance. All of that is real. All of it matters. But underneath the behavior, there may be a genetic floor to the advantage. One that was there before Joseph Smith wrote down the word of wisdom, before the church existed, before the religion had a name. A variant that has been in some of these matrilineal lines for 40,000 years, passing silently from mother to daughter, through migrations and winters and crossings that had nothing to do with Utah and everything to do with survival. The women who carried it to the Salt Lake Basin in 1847 had no idea. The women who carry it today, some of them watching this right now, almost certainly don't either. Cache County, Utah, 1923. A church genealogical record notes the death of a woman, Emma, age 47. Caused cancer of the breast. Her mother died the same way at 43, her grandmother at 49. Her great-grandmother, on the original Pioneer Trek, is recorded only as died young, female complaint. Her great-great-granddaughter, born in 1981, will be the first woman in that line to receive a BRCA1 genetic test before her 40th birthday. She tests positive. She has a prophylactic mastectomy. She survives. The Utah Population Database contains the records of over 2.5 million individuals connected across at least 3 generations of genealogical data. Studies using that database have identified familial clustering of breast cancer across Utah pedigrees extending as far back as 6 generations. That depth, 6 generations, is not a bonus feature. It is the entire point. Below 6 generations, hereditary cancer risk disappears into statistical noise. At 6 generations, it becomes visible, unmistakable. The database was built as a record of faith, kept as an act of devotion to ancestors. But here's what happened. Without anyone changing its purpose, without anyone redesignating what it was for, it became the most powerful oncological research instrument in human history. Nobody made that decision. It was already what it was. Memory, it turned out, was exactly what cancer research needed most. Deborah Necklason, an instructor in oncological sciences at the University of Utah, put it plainly. When they studied these families for colon cancer, she said they could rule out much of the cancer caused by environmental factors. They had, in her words, a much cleaner data set. The no smoking rule in Utah's LDS population, the word of wisdom, that 1833 religious covenant, had been filtering out one of the most confounding variables in cancer genetics for over 150 years before anyone realized it was doing that. 5 generations of women dying of breast cancer, recorded in church ledgers, became the instrument that located the gene. The 5-generation pedigree that made BRCA1 findable was not a research cohort, it was a family. The women in it did not sign consent forms, they were dead. Their descendants, alive, willing, showing up at family reunions with their sleeves rolled up, were participating in research whose implications for their own bodies they could not yet know, because the gene had not yet been found. There is emerging evidence that certain patterns of early life loss, not the deaths themselves, but the anticipatory behavior around the possibility of death, may leave epigenetic marks in descendants of high mortality populations. The Mormon cancer pedigrees are, in one reading, a record of hereditary loss. In another reading, they are a record of hereditary resilience. The question of which framing is more accurate hasn't been settled, and I have to stop here for a second. In a recent episode, we looked at a community whose genetic data was studied for decades without their full understanding of where the research was going. Different faith tradition, different continent, different century, and yet almost the same structure. So let me ask you this. Do you think a community's religious practices can actually change their genome over time? Or does the genome only shift when survival itself is at stake? Drop it in the comments. I read them. Because the next part of this story depends on which one of those you believe. Mark Skolnick arrives at the University of Utah in the 1970s with an unusual idea. He is a population geneticist, and he has noticed something that almost no one else in his field has thought to look at. The LDS Church has been keeping genealogical records for a century and a half, not for medical purposes, but for posthumous temple rites, the religious practice of tracing family lines back through history. Skolnick begins meeting with LDS families. He is invited into their homes. He is shown the church records, the handwritten lists of ancestors, the family Bibles. He is not, at this point, thinking about patents. By the early 1990s, his lab at the University of Utah is one of several globally racing to pinpoint BRCA1 on human chromosome 17. The competitors include teams in Boston, London, Paris. They have resources. They have institutional backing. What Skolnick has that none of them have is a family. 17 women across 4 generations with documented breast cancer, all descended from a single 19th century Utah pioneer couple, all traceable through the Utah population database. That family handed his team the chromosomal address. Science confirmed it. On October 7, 1994, the discovery was published in Science. When the gene was found, the family was told by phone, "Stay with me, because this next part is the reason this video exists." Within months of that phone call, Myriad Genetics was incorporated. Within a decade, it held U.S. monopoly patents on both BRCA1 and BRCA2 testing. In 2013, the Supreme Court heard Association for Molecular Pathology v. Myriad Genetics, a case about whether a company could patent the human genome. The court ruled that natural human genes cannot be patented. The case listed its plaintiffs: medical associations, researchers, patient advocacy groups. The LDS families whose pedigrees had located the gene were not among them. Their interests, whatever those interests might have been, whatever they might have wanted, were not adjudicated. Here's the question I keep coming back to. Skolnick is not a villain. He is not easy to condemn. He did what scientists do, found a research population with remarkable properties, built a tool from those properties, and then, in 1994, did exactly what the American biotech ecosystem was designed to reward. The moral weight here is not on one person. It is on the entire architecture of biomedical research, on a system that was never required to ask whether the people who made a discovery possible had agreed to the terms. Nobody asked. Nobody had to. That was the design. Myriad Genetics is a publicly traded biotechnology company. Its primary asset, the ability to test for BRCA1 and BRCA2 mutations. By 2013, the year the Supreme Court ends its patent monopoly, Myriad has generated over $4 billion in cumulative revenue. The BRCA test is now used by millions of women globally. Every single one of those tests descends in a traceable and documented line from the genealogical records of a community that did not receive a cent of that revenue. The Cancer History Project publication from 2021 documented it specifically. The LDS genealogical records, the Utah population database, the Mormon family's 60% participation rates. These were not supplementary context for the BRCA1 discovery. They were the necessary instrument. Without them, the gene does not get found in Utah. Without Utah, it may not get found in the 1990s at all. The Huntsman Cancer Institute, which was built in part on the commercial success of the era the discovery made possible, states that researchers at the University of Utah can claim the discovery of 50 genes tied to inherited disease risk. 50 from one database, built on one community's love of their dead. Everything I just told you, hold on to it, because it's about to mean something completely different. Here is the balance sheet. Column 1, what was given? 5 generations of cancer deaths recorded in church ledgers. 7-generation family pedigrees maintained as a religious obligation. A non-smoking behavioral baseline, kept faithfully since 1833, that made hereditary cancer signals visible to science. 60% research participation rates at family reunions. Decades of cooperation with an institution that had no formal obligation to account back to the community it was drawing from. Column 2. What was received? The knowledge that their grandmothers had died of something that could now be tested for. Whether that is sufficient compensation is a question the balance sheet does not ask. No one made it ask. Here's what the patent filing for BRCA1 contains if you search it for specific words. Utah, Mormon, LDS, Church, genealogy, family records, 0 results for each of those terms. Not reduced. Not acknowledged with a footnote, 0. The cancer gene that has saved millions of women's lives was found because Mormon families handed scientists their dead. 5 generations of women whose deaths were written down in church ledgers because forgetting them was unthinkable. Science took that grief and built an industry. The community received no royalties, no acknowledgment in the founding documents, no share of the enterprise that was incorporated within a year of the discovery. What they were given was the knowledge that their grandmothers hadn't died for nothing. Whether anyone told them to frame it that way, that too is not in the record. The standard framing targets Myriad Genetics as the entity that commercialized the human genome, but Myriad didn't extract the data under false pretenses. The families gave it. They showed up. They rolled up their sleeves at family reunions and said yes. The question isn't whether anyone lied. The question is whether giving freely without knowing the commercial destination of the gift constitutes consent in any meaningful sense. That question is not about one company. It is about the entire architecture of how biomedical research draws from communities that trust it, and it has not been answered. UCLA 1979. 2 researchers, James Enstrom and Lester Breslow, begin a study. They are not from Utah. They are not LDS. They are asking a simple question about lifestyle and lifespan, following actively practicing Mormon church members in California for 25 years. The results, published in Preventive Medicine in 2008, are still among the most striking longevity figures ever documented for an identified American population cohort. Mormon men who don't smoke, attend church weekly, have at least 12 years of education, and are married. Life expectancy 84.1 years. That's 9.8 years longer than comparable U.S. white males. Mormon women, 86.1 years, 5.5 years beyond comparable U.S. white females. At the time of publication, these were the longest documented life expectancies for any identifiable group in American medicine. What they found next changed everything they thought they knew about this. The behavioral explanation, no tobacco, no alcohol, social cohesion, religious structure, accounts for most of the difference. That's real, undeniable. But underneath it, the mitochondrial K2 data from segment 3 sits and refuses to leave. If some portion of the longevity floor is genetic, if the advantage predates the word of wisdom, predates the religion itself, then this is not purely a story about what faith does for health. It is a story about what a community is, biologically, before it decides what it believes. After everything I told you in the last segment, after the balance sheet, after the patent search with 0 results, this finding no longer reads the same way it would have read 20 minutes ago. The community gave science its dead. In exchange, not by any negotiation, not by any design, they also gave science the most valuable natural longevity study in modern medical history. Their bodies were the data. Their dead were the instrument. Their faith was the preservation mechanism that kept the records intact and the confounders out of the analysis. The Utah data confirms the California data. LDS versus non-LDS Utahns show consistent mortality differentials across all major cause of death categories. The behavioral explanation holds. But the genetic floor was there before the behavior began. And the question of whether a community that contributed all of that, their dead, their records, their cooperation, their bodies, received something commensurate in return has, finally, one partial answer. They lived longer. Whether that was ever the deal, no deal was struck. No one sat across a table from these families and said, "Give us 5 generations of your grief, and we'll give you 10 extra years." It happened. The exchange was real. The terms were never disclosed because no terms were ever agreed. And here is something that has not been formally studied as of any published research available today. The 1847 Mormon Exodus, 70,000 people walking to Utah across 1,300 miles of unmapped American territory, with hundreds dying before they arrived, filtered the founding population. The survivors were not a random sample of 19th century American Protestantism. They were selected by geography, by commitment, by age, by the physical endurance required to complete that crossing and stay. Whether that filtering left a measurable genetic signature, whether the longevity advantage is, in any part, a survivor effect of 1847, has never been formally investigated. Winter, 1846 into 1847. The main Mormon pioneer company is encamped at Winter Quarters, Nebraska, now the city of Omaha. The official church records documents 600 deaths during that winter alone. 600 people. In one encampment, in one season. The survivors left in the spring. Their ages, their family relationships, their health at the time of departure. These are, in many cases, traceable through the genealogical records that would later form the Utah Population Database. This is not a metaphor for a genetic bottleneck. It is a documented instance of one. There are three things a 1984 study by McClellan and colleagues at the University of Utah found when comparing Mormon gene frequencies to other religious isolates: the Amish, the Hutterites, the Mennonites. The third one is the one that changes everything. The Amish and Hutterites show measurable genetic drift from their ancestral European populations. The Utah Mormon population does not. Their gene pool tracks closely enough to their northern European origins that the founding size and subsequent gene flow appear to have prevented meaningful drift. But, and this is the part that has never been followed, the specific subset of northern European haplogroups that passed through that 1847 crossing, including potentially the K2 mitochondrial subclade, has never been tested for bottleneck effects. The bottleneck has been acknowledged. The bottleneck has never been measured. Now trace the chain backward. The BRCA1 discovery in 1994 traces to multi-generational Mormon pedigrees. Those pedigrees trace to the Utah Population Database. The database traces to LDS genealogical records. Those records trace to the pioneer survivors of 1847. And the religious obligation to keep those records traces to a single document written in 1833. Section 89 of the Doctrine and Covenants, known as the Word of Wisdom. A spiritual covenant received as a revelation that told members to honor the body by avoiding tobacco, alcohol, and harmful substances, and to eat a balanced diet. That document was not a health guideline. It was received as something sacred. The men and women who followed it did so because they believed in it, not because they knew it would make their cancer death legible as genetic signal to researchers 150 years later. Not because they knew it would allow oncologists to strip out behavioral noise and see the hereditary pattern underneath. They followed it because it was the right thing to do. One document, written in 1833, 2 centuries of consequence. An industry built from the compliance of people who had no idea what they were complying into. The chain is not a metaphor. Every link is documented. And at the end of that chain is a research gap that, as far as the published record shows, no one has proposed closing. A formal, rigorous comparison between the 1847 founding cohort's descendants and the northern European populations they came from. The bottleneck and its genomic consequences measured at last. Nobody has funded that study. Nobody has proposed it publicly. Nobody has asked the families if they'd like to know the answer. What has been measured is this. The Utah Population Database contains the medical and genealogical records of 8 million people. Researchers at the University of Utah have used it to identify over 50 genes tied to inherited disease. Every major hereditary cancer gene confirmed in the second half of the 20th century, BRCA1, BRCA2, APC for colon cancer, P16 for melanoma, KCNH2 for cardiac arrhythmia, has a traceable connection to this database. The database was built on LDS Church genealogical records. The church records were built on faith. That chain has a last link, and it connects to you. If you have had a genetic cancer test, or if someone you love has, the chain of causation running backward from that result passes through a religious community's love of their dead. If you carry haplogroup K2 in your mitochondria, there is a specific matrilineal database of 14,900 Utah lineages that helped science identify what it means. The question of what you owe for that, and to whom, is not a rhetorical question. It is the exact question that the legal system, the bioethics community, and the research community have not yet answered in a way that reaches the families it actually concerns. The law addressed the gene patent. The Supreme Court ruled. The families were not plaintiffs. The question of whether a community's genealogical generosity constitutes an intellectual contribution to a commercial product, that question has no docket number. It has no pending ruling. It has no institutional home. If this episode changed the frame through which you understand genetic research, consent, and what communities give to science without being asked, share it with someone who has received a genetic health test. Subscribe if you want to stay inside these questions, because the one I'm about to leave you with has no clean answer, and clean answers in this field are usually the ones that are wrong. The families in the Utah Population Database did not set out to save your life. They set out to remember their dead. The fact that these turned out to be the same act is either the most extraordinary coincidence in the history of medicine, or it is evidence that the way a community organizes its memory has consequences that reach forward in time further than anyone inside that community could ever see.